It's possible that the brain is the most complex and unknown organ in the entire human body. How exactly does this clump of cells give rise to memory, emotion, and cognition? This is a question that scientists have been wrestling with for a while now, and recently a new tactic has come up to answer it: mind control. No, really. We like to think about the brain like a computer, and in some ways it is, but it's also even more complex than any computer we've been able to design so far. Each of our neurons passes electrical signals down its axons into neighboring cells. Differences in voltage across the membrane propagate this signal. Normally, this kind of response and action potential occurs because of a stimulus from a nearby cell. But what if we could bypass the stimulus and excite the cells ourselves? This is the premise of optogenetics. By switching neurons on and off with light, we can understand exactly what they do and cause animals to do things like wake up, run in circles, and by targeting the right cells, cure blindness. This is how it works. By taking light receptors called rhodopsins from prokaryotes that function as ion channels and pumps, we can genetically engineer switches onto neurons so that we can turn them on and off as we wish. To make this happen, scientists have to remove the plasmid from a type of green algae who uses rhodopsins to sense light so it can photosynthesize. The genetic information coding for rhodopsin is put into the nucleus of an embryonic stem cell that will eventually differentiate into a neuron using a retrovirus. Retroviruses are a kind of virus that insert their genetic info into host cell DNA. Scientists use this as a tool in genetic engineering to insert genes into certain cells. We can also insert genetic information into cells using non-viral methods like transfection. Channel rhodopsin is the type of opsin used to switch neurons on in the presence of blue light. Other opsins, called ochrorhodopsin and halorhodopsin, can also be used to turn neurons off or inhibit signal transduction in the presence of yellow and green light. By inserting a light into the skull, we can illuminate the entire brain, and only the cells with the rhodopsin light switches will be activated. With this tech, we can determine what different neurons do and work towards being able to better understand neurological diseases and disorders so that we can one day treat them effectively. Our understanding of the applications of this tech is only growing, and it will be exciting to see where optogenetics takes us.